Welcome to your first semester of cell biology. Today, we are going to explore one of the most fascinating topics in eukaryotic cells, semi-autonomous organelles. Imagine a tiny, specialized factory operating inside a much larger city. It has its own rule book and its own workers, but it still relies on the city's main power grid and supplies to survive. This delicate balance of independence and reliance is exactly how certain organelles function inside your body. By the end of this video, you will understand what makes them unique, how they operate, and why they are absolutely critical to human health and evolutionary history. Let us dive in. To understand our topic, let us start with a simple analogy. Imagine a massive, global corporation that decides to purchase a smaller, highly specialized startup company. The startup is allowed to keep its own internal rulebook, its own specialized workers, and manage some of its own daily tasks without asking for permission. However, it still relies heavily on the parent company's headquarters for its main budget, major supplies, and overall direction. It is not completely independent, nor is it completely controlled. It exists in a state of being semi-independent. This is exactly how semi-autonomous organelles, like mitochondria and chloroplasts, function inside the cells of your body. They are special compartments that have a little bit of their own independence, but still rely on the main cell to survive. Now that we have our analogy, let us look at the actual biology. In cell biology, semi-autonomous organelles are specialized subcellular structures that possess their own genetic material and protein synthesizing machinery. Unlike other organelles, such as the Golgi apparatus or lysosomes, which are entirely built by the cell's main factory, mitochondria and chloroplasts contain their own circular DNA and their own ribosomes. Because they have these tools, they can replicate themselves independently of the main cell cycle through a process very similar to bacterial division. However, they are termed semi-autonomous rather than fully autonomous because their small genomes only encode a tiny fraction of the proteins they actually need to function. The vast majority of their proteins are encoded by the cell's main nuclear DNA. Understanding this unique structure leads us to why these organelles are so important in modern science. The semi-autonomous nature of these organelles is absolutely critical for modern medicine, genetics, and evolutionary biology. Because mitochondria have their own distinct DNA, mutations in this specific genetic code lead to a unique class of metabolic diseases known as mitochondrial myopathies. These diseases primarily affect energy-hungry tissues like muscles and the brain. Furthermore, because mitochondrial DNA is inherited almost exclusively from the mother, it allows scientists to trace maternal ancestry back thousands of years. This helps us map human evolution and ancient migrations. Without this unique genetic separation, we would not have this powerful tool for genetic tracing, nor would we understand why certain inherited diseases completely ignore standard genetics. This biological balance brings us to a mathematical way to represent genetic autonomy. To truly understand what makes these organelles only semi-autonomous, we can look at the balance of where their proteins come from. We can represent the total protein requirement of the organelle as a simple sum. In this equation, P total represents the total number of unique proteins required for the organelle to function properly. The variable PMT stands for the number of proteins that are directly encoded by the organelle's own mitochondrial or chloroplast DNA. Finally, pNuc represents the number of proteins encoded by the cell's nuclear DNA, which must be imported. If the value of PMT were to decrease over evolutionary time, the organelle would become more dependent on the nucleus, requiring an increase in p-nuc to survive. Building on that total protein equation, we can now measure exactly how independent the organelle is. We do this by finding its percentage of genetic autonomy. Here, the variable A represents the percentage of genetic autonomy possessed by the organelle. We calculate this by taking the organelle-encoded proteins, PMT, dividing it by the total proteins, P total, and multiplying by 100 to get a percentage. This formula gives us a clear, quantitative way to measure independence. If the number of proteins encoded by the organelle's own DNA increases, the value of A will increase, meaning the organelle is more self-sufficient. Conversely, a lower value of A indicates a heavy, almost total reliance on the host cell's nucleus for survival. Now that we have our formulas, let us apply them to a real-world biological scenario involving a human cell. A human mitochondrion requires approximately 1,500 different proteins to perform all of its functions. However, its genome only encodes 13 of these essential proteins. We need to determine exactly how many proteins must be imported from the nucleus. By rearranging our first formula, we isolate the unknown variable for nuclear proteins. We subtract the 13 proteins made by the mitochondrion from the 1,500 total proteins required. This simple subtraction reveals the massive hidden workload of the host cell. Exactly 1,487 unique proteins must be transported across the mitochondrial membranes just to keep the organelle functioning. Having found the import burden, let us calculate the actual percentage of genetic autonomy for this human mitochondrion. We want to find out how genetically independent the mitochondrion actually is. We calculate the autonomy percentage by dividing the 13 internally produced proteins by the 1,500 total proteins. Multiplying the result by 100 converts our decimal into a readable percentage. The final calculation reveals that its genetic autonomy is a mere 0.867%. This mathematical step translates raw protein counts into a conceptual measure of independence. This incredibly low percentage perfectly illustrates why we use the prefix semi in semi-autonomous. It heavily depends on the nucleus for over 99% of its functional protein needs. Now, let us compare our previous result with a different semi-autonomous organelle, the chloroplast found in plant cells. In a certain species of plant, the chloroplast requires a total of 3,000 proteins to carry out photosynthesis. The chloroplast's own circular DNA encodes 120 of these proteins. Our strategy here is identical to the first example. Once again, we subtract the internally encoded proteins from the total required proteins. Taking 120 away from 3,000 leaves us with 2,880 proteins that must be imported. This shows that the plant cell nucleus is responsible for manufacturing a massive amount of material specifically for the chloroplast, requiring highly efficient transport mechanisms. With the nuclear protein count established, let us find the autonomy percentage for the chloroplast to compare it with the mitochondrion. We find the autonomy percentage by dividing the 120 chloroplast-encoded proteins by the 3,000 total proteins. Multiplying by 100 gives us the final percentage of independence. Our results show that its calculated genetic autonomy is 4.0%. Notice how the larger number of internally encoded proteins affects the numerator of our fraction. This is significantly higher than the human mitochondrion's 0.867%. While the chloroplast is slightly more genetically autonomous, it still overwhelmingly relies on the plant cell's nucleus for 96% of its needs. These mathematical examples bring us to a clear visual representation of this dual genetic system. On screen, you are looking at a schematic of a eukaryotic cell that perfectly summarizes everything we have discussed. On the left, you see the cell's nucleus. Notice how nuclear DNA is transcribed into messenger RNA, which travels out into the cytoplasm. There, a ribosome translates it into a protein labeled p-nuc, which is then imported into the mitochondrion. On the right, inside the mitochondrion itself, you see its own circular DNA producing a protein labeled PMT. Finally, look at how both of these proteins converge. They join together to form the functional mitochondrial complex. This diagram beautifully demonstrates the delicate balance of power and dependence that defines semi-autonomous organelles. As we wrap up, let us review the most important ideas from this topic. First, remember that semi-autonomous organelles, like mitochondria and chloroplasts, operate like a cell within a cell. They retain their own DNA and ribosomes, allowing them to produce a small but vital fraction of their own proteins independently. However, their massive reliance on the nucleus for the vast majority of their components is what makes them only partially independent. This unique dual genetic system is the absolute cornerstone of cellular energy production, and it provides a fascinating window into the evolutionary history of complex life. You have now mastered the fundamental concepts and mathematics of semi-autonomous organelles. Great job!